Hello. Today we discuss uh, questions of the cellular transport. It is topic of the lecture number 2. As we know, cells of the human body live in a carefully regulated fluid environment. The fluid inside the cells we can call like intracellular fluid. Intracellular fluid. It is intracellular space that filled with intracellular fluid. For the average person with a body weight near 72 kilogram, this body contains approximately 42 liters of the water. It is around 60% of the body weight. And these body fluids are separated into the two main portions. First portion is intracellular space. We can call this like intracellular fluid. It is around 25 liters. And extracellular space. Extracellular space is divided into the several parts. It is interstitial fluid, it is around 13 liters for average person. Of course, intravascular liquid like blood, around 3 liters, and another intravascular liquid is lymph, 1-2 liters volume. And the last one, so-called transcellular fluid. Transcellular fluid. Total volume is very low, is around 1 liter, and it is fluids such as cerebrospinal fluid, synovial fluid, fluid of the fluid of the fluid of the spaces that separate some like visceral organs and wall of the thoracic cavity. It is space, small space between these sheets that filled by the film of the fluid. It is very small volume of the fluid. Okay, so and between these compartments of the body, we can find exchanging of the water and the solutes. The barriers that separate these two compartments, extracellular and intracellular, are the cell membranes, actually. It is cell membranes. The body must maintain volume and composition of the intracellular and extracellular compartments. Such regulation is the result of the transport across cell membranes. And today we discuss how cell membranes regulate distribution of the ions and water between intracellular and extracellular compartments. So if we want to discuss questions of the transport through cellular membrane, we need to firstly discuss components of the membrane that can make this transport. So main part of the membrane is phospholipid bilayer. And inside this phospholipid bilayer we can find some integral proteins, and like one of the variant of this protein is pore in membrane. It is a water-filled hole in membrane that can pass component. We can call this like pore or channel. Channel or pore. So in some cases, substances can pass directly through the phospholipid bilayer. In another case, substances that cannot directly pass through this structure, it needs specific hydrophilic pore inside membrane. Okay. Next variant of the transportation, next structure, molecular structure for passing of the substances is another kind of integral protein that cannot produce hydrophilic pore inside, but by own conformational changes can transport substances through membrane, so-called transmembrane both-directed transport without channel, by own conformational changes. Okay, so continue. Membrane, another integral protein. Another integral protein that not produce channel 2, but for working of these elements we need energy of ATP. This transport required energy of ATP. It is directed transport, direction of this against concentration gradient. Against concentration gradient. And we can call this transport like active. So required energy. Energy required transport we will call like active. This energy used from ATP molecules, or it is most often substance but maybe another like GTP, etc. Okay, so we can combine these transport transport mechanisms using carriers. Using carriers, it means no hole inside, no water filled inner pore. So if this transport mechanism goes in direction of the concentration gradient, we can call this like facilitated diffusion. And direct transport through cell membrane component, through phospholipid bilayer, or through the channel or hydrophilic pore. It is maybe ions like sodium, potassium, maybe water, and another water soluble components. We can combine this mechanism in simple diffusion. Okay, so simple diffusion and facilitated diffusion both are passive mechanisms. And one of the passive is diffusion, not required energy of ATP. So, and now we discuss these transport mechanisms separately. So we start from the first is diffusion. How we can define diffusion? Diffusion process is transport or transmission of the substance as a result of random heat movement or motion, of molecules or ions in all possible directions. And we can call this process like Brownian motion. Okay, so diffusion is a transport of the substance as a result of the random heat motion of molecules or ions in all possible directions. What is the driving force for this action, for this movement? Driving force is gradient of the concentrations. Gradient of concentration. Okay, so if we have two places of the solution that has differences in concentration of the solute particles, these particles start to move from places of high level of concentration to low level of concentration. And as a result of this process, after time, we receive equal concentration of the solid particles in all parts of the solution. In this case, concentration C1 in first place, C2 become equal. So how to describe process of the diffusion and what factors influence to the effectiveness of the diffusion process? One of the main parameters that indicates effectiveness of this process is the coefficient of diffusion. Coefficient of diffusion. Coefficient of diffusion. Coefficient of diffusion describes with help Stokes-Einstein equation. Stokes-Einstein equation that tell us that coefficient of the diffusion depends upon temperature, depends upon viscosity of the solution, viscosity of the solvent part of the solution, and radius of the substance that diffuse in this solution, radius or size of molecule, radius of molecule, radius of molecular or ion. So, and this equation indicates this ratio, like ratio between gas constant and temperature. Avogadro number 6 pi, radius and viscosity. We can see that more temperature, the more diffusion process, and inversely proportional to the size and viscosity of the solution, size of the particle for diffusion and viscosity of the medium. Diffusion coefficient, it is part of the law that describes diffusion process in the solutions. This law we can call like first law of diffusion, that formulated by Fick. It's Fick's law. First Fick's law about diffusion. Fick's law describes how much molecules of the solid particles will be diffused in the solution upon time. So in other words, rate of diffusion is proportional to amount of the diffusion of substance upon time. Rate of diffusion, in our case, depends upon several factors, including coefficient of the diffusion. So, in this equation, J is rate of the diffusion, A is surface area, the diffusion coefficient, delta C is gradient of concentration, and delta X is the distance. Distance of diffusion. So, as we know, diffusion process can happen like directly through the cell membrane, through phospholipid bilayer, directly, or using pores of the membrane, that filled by the fluid, filled by the water, and this process can be easier for the hydrophilic. Okay, in this case, concentration difference is the difference in concentration between two solutions separated with a cellular membrane. And if we have difference of this, some of the concentration is greater, we have directed process. Diffusion in this case goes in places from places with high level of concentration to the places with low concentration. This distance is the X, how much distance travel substance from one place to another, and A, it is surface of the membrane, surface area of the membrane, or number of the channels in membrane that will be used for the diffusion. So what factors influence to the diffusion through cell membrane directly? So diffusion through lipid bilayer. Diffusion through phospholipid bilayer depends upon nature of the substance, presence of the charge of the object. What means nature of the substance? We know that substances differently can dissolve in different solutions. we can divide all substances into the water soluble and lipid soluble. We can call this like lipophilic or hydrophilic substances. So the more lipophility of the substance, the faster process of the diffusion. What factors influence to the ability of the substances to dissolve in the lipids of the phospholipid bilayer? First factor is polarity. Polarity of the substance. Polarity of the substance, it is property when charge not equal distributed on the surface of the molecule, okay, or maybe equal. In polar substances, we can see this difference in the collection of the positive and negative charges on the surface. Like water molecules, it is polar. Water molecules. And non-polar molecules like urea, glucose, respiratory gases, oxygen, carbon dioxide. Okay, so polar molecules usually cannot transport directly through the phospholipid bilayer, because molecules cannot penetrate between molecules of the phospholipids on membrane. Non-polar molecules, transport of non-polar molecules depends upon presence of the specific channels. In case of the urea and glucose, because phospholipid bilayer for these substances none possible if we talk about high molecular weight like urea or glucose. But oxygen and carbon dioxide can very easily pass through this. Okay. I'm sorry. For some mistake, actually, urea and glucose, it is polar substances like water, but greater molecular weight. Urea, glucose, and non-polar substances, for example, respiratory gases and like hormones of derivatives of the cholesterol. Cholesterol derivatives like testosterone, estrogens. Okay. So we can conclude that polar molecules very hard to pass through the phospholipid bilayer directly. For this transport needs specific proteins of membrane, like aquaporins for water, like GLUT transporters for the glucose molecules. For non-polar urea, of course, can be transported through the aquaporins too, but possibility of this transportation less than for water. It is in 100 times less than for water, but possible. Non-polar substances can easy to dissolve in the phospholipid bilayer and transport to the own concentration gradient in a specific direction. So as we conclude about transportation, not only nature of substance can influence, it is maybe molecular weight. Molecular weight. Okay, so for water less, more effective. For urea, for glucose greater, less effective. Okay, so next property, presence of the charge. Charge is a property of the ion. As we know, molecular is electron neutral object. If this object lose one electron or receive additional charge, it receive positive or negative charge. All charged objects very hard transport through the phospholipid bilayer directly, because in solution these objects are surrounded by the polar molecules of the solvent. In our body it is water, and this water coverage of this object not allow penetrate through the molecules of the phospholipid bilayer of our membrane. And coefficient of the diffusion of these substances is very low. For the transportation of these charged objects, for the ions, we need to use specific proteins, specific transmembrane proteins with name ion channel. We can conclude that diffusion of the substances across cell membrane directly through the phospholipid bilayer or using specific protein molecules with hole inside, all this transport calls simple diffusion. Simple diffusion. Simple diffusion. Another type of diffusion that can use carriers of cellular membrane. These transport mechanisms calls like facilitated diffusion. Facilitated diffusion using carriers. So, how work this mechanism of the transportation? These integral proteins of membrane that move this transport have no any watery channel inside. It is not pore. And transport happens like result of the conformational changes of the protein molecules. And the moment of these conformational changes, this protein transport substance in direction of the concentration gradient from one side of membrane to another. On this molecule usually present active site after connection of the transported molecule. On this active site, shape of the protein changes, and molecule or ion will be traveled to the another side of the membrane. We have some variants of the facilitated diffusion. In some cases, this protein carrier transport only one molecule. Such transport we can call like uniport. Uniport, transport of single substance, single object. Second one is transportation of the two substances simultaneously, but in one cases this transport happens in same directions for both transported molecules. This kind of transport we can call like symport. Symport. So on the picture I will show you this transportation. Okay, symport, two different molecules, two different maybe ions transported simultaneously. After connection to the two active zones of the both substances, start conformational changes, and these substances transmits to the another side of the membrane. So this transport we call symport. Third variant of the transportation is antiport. Antiport. In this case, two transported objects transported into the both transported into the opposite directions, relatively membrane. In this case, on protein molecular surface, we have two active zones located in different surfaces of the membrane. One in the intracellular space, another in extracellular space. And in the moment when transported materials connected with these active zones, start conformational changes, and two substances go in opposite directions. One inside the cell, another one outside the cell. This kind of transport we can call like antiport. These variants of the facilitated diffusion. What are the differences we can call for this transport in difference with simple diffusion? What are the properties? Okay, so first of all, transport facilitated diffusion is saturated transport. Saturation. How it happens and why? If we see to the graph of the rate changes accordance concentration gradient changing from 0 to the high concentration gradient, we can see that simple diffusion has direct ratio with concentration gradient. The more concentration gradient, the more rate. The more rate. For the and we have no limit in the increasing of the rate of diffusion. This ratio goes to infinity. The more concentration gradient, the more rate. But for facilitated diffusion, we have specific moment. And graph of the facilitated diffusion looks like this. We have saturation effect. When all elements of the membrane that can transport this material through membrane are busy by the transport process. We cannot increase to a new level, to new step of this substance. No molecules, no additional molecules for the connection of the new molecules. All transport carriers are busy by transportation, and we cannot increase the rate of diffusion by the increasing concentration gradient. But on first moment of diffusion, when number of molecules of the carrier molecules bring new molecules of the transported material, we can see that rate of diffusion is very high in comparison with simple diffusion. On first moments, it is higher, it is more effective, but till moment of the achievement of this maximum level, this transport fully saturated and further increasing of the concentration gradient not influence to the effectiveness of the transportation. Next property of the facilitated diffusion, it is competition. Usually, or sometimes, one carrier can transport several types of molecules that has similar molecular structure. And if a carrier bring two molecules simultaneously, these two molecules start to compete to the carrier, and concentration of one substance in this case influence to the effectiveness of the transportation another substance. Okay, if we have two substances, X1 and X2, if concentration of the concentration gradient of X2 increases, it will increase rate of diffusion for X2 molecules, but in this moment, X1 molecule not enough for the same rate of the transportation, and this rate, of course, decreases. This property calls like competition for the carrier or competi inhibition of the transportation. So we can conclude that facilitated diffusion and simple diffusion are the passive kind of transport. Facilitated diffusion and simple. Next kind of the passive transport, it is osmosis. Osmosis. What is osmosis? Osmosis is a movement of the solvent molecules through the semi-permeable membrane from low concentrated solution to high concentrated solution. Osmosis, it looks like a simple diffusion, but in this case, we talk about diffusion of the water through semi-permeable membrane. Why we need to discuss osmosis in case of the biological transport? Because our membrane is selectively permeable for the substances, and we know that in a resting membrane of the usual animal cell, we have a large amount of the pores. We know these like aquaporins, specific channel for the transportation of the water. There are several types of the aquaporins we know, and localization of these molecules differ in the organism. One kind we can find in the kidneys, and it is aquaporins that can be regulated by the hormones, for example, EDH in this case. And other aquaporins are present in the intestine, in the liver, and in other organs. Most of the membranes of our body contains these molecules. That's why membrane highly permeable for water usually. And phospholipid bilayer, as we know, impermeable for the most solute particles, such as any inorganic molecule that in solution position of this substance usually in ionized form. For example, sodium chloride in solutions dissociates into the sodium ions and negatively charged chlorine ions. Sodium and chlorine cannot pass through the phospholipid bilayer because brings charge, and this object cannot be transported through the phospholipid bilayer, and that's why osmosis happens in cases when we have semi-permeability like this, when membrane is permeable for water, for solvent, but impermeable for the solute particles like ions in our case, like proteins. Starts process of the solvent movement through the membrane in direction of the own concentration gradient. If we talk about concentration of water in solution, about concentration of the solvent, we can define this process like movement of the water from places with low concentration, with high concentration of the water to the places of low concentration of water. So, one of the main characteristics of effectiveness of osmosis we can call like osmotical pressure. Osmotical pressure. What is osmotical pressure? What is osmotical pressure? If we take beaker like this, separated with semi-permeable membrane, that permeable for water but impermeable for the ions, impermeable for the ions, and we fill this beaker with solution, with two solutions. One solution with high concentration of the solid particles, and another part of the beaker filled with pure water. In this system starts process of the water movement through the semi-permeable membrane. Water movement. And after time, we can see some changes that happens in the system. After time, level of the fluids in these parts of the beaker will changes. In left part, level will be higher and concentration become more diluted than previously. And on this system will be created pressure, some pressure to the membrane from this direction. For the measuring of this pressure, we can use opposite method. Osmotical pressure defined as force, external force that we need to apply to the surface of the solution to prevent osmotical movement of the liquid from low concentrated solution to high concentrated solution. This pressure we can call like osmotic pressure. What is significance of the osmotical pressure in our body? As we know, in our body we have compartments, intracellular compartment and extracellular compartment. And osmotical pressure influence to the transportation of the water between these compartments. Usually, in normal conditions, we have 290 to 300 milliosmol of the osmolarity of the internal fluids. We can call this like isotonic or isoosmic condition. Isotonic condition. It means when cell has surrounding osmolarity approximately same as intracellular, it means we have optimal water exchanging between intracellular and extracellular spaces, intracellular fluid and extracellular fluid. So in this case, we have optimum water balance between these two compartments. But if cells will feel high concentration of the extracellular fluid, now this condition we can call like hypertonic. Hypertonic conditions. What happens in hypertonic condition? In hypertonic condition, activates osmotical transport of the inner fluid, and cells start to shrink because lose own water. Water goes from low concentrated solution to the extracellular fluid, and volume of the cell decreases. It will influence to the tension of the cellular membrane, and it is very powerful mechanical force. And the opposite case, hypotonic. Hypotonic solution. In hypotonic solution, we can see opposite picture. Volume of the cell start to increases because osmosis will move water inside the cell, and the volume of the cell in first moments increases. Our cells swelling, and this process finally can cause bursting of the cell. Especially it important for the blood cells, because water exchanging between intracellular fluid and extracellular fluid for red blood cells, for RBCs, is equivalent to the 100 volumes of the inner fluid per several seconds. It is very fast. If RBCs feel non-optimal tonicity of the extracellular fluid, of the blood plasma, it is immediately influenced to the volume of the cell. This cell has resistance to these changes, but after achievement of the limit of this resistance, RBCs can burst. And after bursting, RBCs release hemoglobin to the solution. Hemoglobin is very useful substance for the transportation of the oxygen, but in solution of the blood plasma, it is toxic substance because influence to the blood flow in the capillaries, especially in the kidneys. Kidneys suffer first time, and blood flow in the internal organs because of this can stop, and this very high amount of the risk for the maintaining of the life. That's why maintaining of the tonicity, it is very important mechanism for our body. We have special physiological system for the maintaining of the water electrolyte balance of our body. Okay, osmosis is one of the type of passive transport, not required energy of ATP, goes passively in direction of the concentration differences, concentration gradient, but it is specific diffusion, specific transport of the solvent, not solute, as for usual simple diffusion, but use the same laws for the transportation like simple diffusion.